Welcome back to another topic of RNA biology. So today we will start with a new section which talks about the RNAs, its transcription, its processing and the life cycle. So this is a structure of the RNA polymerase, which is a protein enzyme which helps in the production of the RNA from the DNA template. It is having the shape of an inverted C and it basically clinches on to the DNA strand. Remember, whenever the transcription takes place, only one strand of the DNA act as the template. During DNA replication, both the strands act as the template, whereas when DNA contributes to the production of the RNA, only one strand of the DNA act as the template. And in this topic, we will cover a few topics as listed here. It's an outline. So, transfer of the genetic information, that we call it as the central dogma. And central dogma basically means that a routine step of functioning is always followed, no matter which organism you are talking about. If it has a genetic material, it has to follow a routine procedure. We'll see more in detail about that. And then the process of gene expression, how the so-called gene expression takes place in a cell. And then transcription in prokaryotes. Prokaryotes are bacteria and other single-celled organism and who doesn't have a proper nucleus. That is what we refer to as prokaryotes. Whereas the next topic is the transcription and RNA processing in eukaryotes. That means those organisms that have got a proper nucleus. Every organism that have got a nucleus is a higher order organism which has got a higher level of or a higher complexity in terms of its gene expression and functioning. And then we also learn about interrupted genes in eukaryotes. Interrupted genes basically means in a gene or a sequence there are wanted as well as unwanted genes, unwanted regions. So we call those genes as interrupted genes. Means in order for these genes to function, we need to get rid of this unwanted portion. Unwanted areas should be removed before this RNA can function as a proper mRNA for the translation. And we also will learn about the removal of the intron sequence by the process called RNA splicing. And the transfer of genetic information, the central dogma in molecular biology, what it basically means is the information is stored in the DNA. We have already learned that in the RNA world hypothesis, the information was generated randomly in an RNA, which is now getting converted into the DNA for the safekeeping purpose. And so this information stored in the DNA should be made available as and when required. So that process has to be done through RNA transcription. So the process of formation of RNA from the DNA, we call it as RNA transcription. And this transcribed RNA now gets converted into protein through a process called translation. And so that is what mentioned here, DNA getting converted to RNA and RNA getting converted to protein. So the DNA to RNA or the information present in the DNA, a character present in the DNA is basically called as the genotype of an organism. And this RNA, when it gives rise to protein, or because the proteins are the one which is going to perform a task, they are the workforces in a cell. So that functioning is referred to as phenotyping. So genotyping and phenotyping can be explained in a simplistic term. Say for example, if you know to draw an artwork, or you know how to sing, you are a classical singer, but whether you sing or whether you perform your artwork depends on the circumstance. So genotype is what? The ability to sing or ability to perform an artwork is the genotype. Phenotype is the one whether or not you will sing or whether or not you will paint. So that is what phenotype. So genotype is there doesn't mean that it has to express the phenotype. Say for example, in your fingertip, the cell, any given cell, if you take from your fingertip or on the root of your hair, has the entire genome of you, entire information required for an organism starting from its development throughout the functioning is present. Roughly, we have around 23,000 genes a human being has. All the genes are there, but they are not expressed. Any given time, around 10 to 12,000 genes are expressed in a given cell. So, what it indicates that the number of genes, that is the genotype, need not necessarily express any given time. Based on demand, based on situation, this happen. And let us think about situation in an RNA virus. So RNA viruses are plenty, COVID virus, influenza virus, many more, like even HIV virus. Many are RNA genome viruses. So they don't have DNA in them. But what they do, they either make a copy of itself, that is an RNA copy produced from the RNA itself, or RNA gets converted into DNA depending upon the nature of the virus. This is how they fall into the central dogma. In any case, once the RNA getting converted into DNA, it has to undergo a process of transcription again in order to have the phenotype of the virus to be expressed. Or say if an RNA is making a copy of another RNA, then it can participate in the protein translation directly, depending upon which virus you are talking about and which host, which organism we are referring to. This may vary. But why we mentioned about virus here is because the central dogma has to be followed strictly and it is unchallenged. And now, when you talk about the central dogma, we can see the transcription takes place from only one strand of the DNA, and that we call it as template strand. And normally a DNA strand, when it is present, it is double stranded and they are complementary to each other. Say in one strand you have got A, then the opposite strand you have got a T, that is thymine. A stands for adenine, T stands for thymine. If one strand has got G, then the opposite strand has C. So what we have to say, they are not identical copy, but they are complementary copy following this rule. A pairs with the T and G pairs with the C. Like that it continues. So that is the double stranded DNA's quality. Same way, if one strand is of the DNA is acting as a template, the A containing region now have an RNA which has got U instead, uracil instead of thymine, because we know thymine is a feature of DNA. Thymine is not present in the RNA. But whereas if a G is present in the template DNA, then there will be a C. C stands for cytosine, G stands for guanosine. So this complementarity rule is followed when a RNA is transcribed from one DNA strand or one DNA template. And this gives rise to primary transcript, and we call it as messenger RNA. And this RNA has to undergo some chemical modification depending upon the organism. Some process are called 5' capping and 3' polyadenylation, and then sometimes if a eukaryote have got interspersing region such as introns, if they are present, they have to be removed, a process called splicing, mRNA splicing, and this is helped by structures called spliceosome, without which you will not be able to make a mature mRNA. And that is what you are seeing here in this right hand side panel. You can say that this DNA, when it is opened up, it is capable of making a copy of itself through a process called DNA replication. For that, it uses an enzyme, DNA dependent DNA polymerase. And now, once it is copied, each strand participates in the production. So, one double-stranded DNA now end up getting two double-stranded DNA, because one cell become two, two become four, like that. So, DNA dependent DNA polymerase helps in the production of DNA copy. Now, another enzyme comes into picture. Remember, these are all protein enzyme. That enzyme is DNA dependent RNA polymerase. So name itself indicates it needed DNA to act as a template, and the product is RNA. So DNA dependent RNA polymerase comes into picture, and it make a copy of RNA, or it transcribes an RNA by making use of one strand of the DNA. And this mRNA, if it is processed properly, it can participate in translation. And this is a complex process that involves transfer RNA, ribosomes, and many other protein factors for the proper termination of the translation, etc. We will see about that more in detail, but end of the day, or end of this process of translation, you end up getting a polypeptide. It can be an enzyme, or it can be a structural protein, can be anything, because remember, the proteins are the major workforces in a cell. And now there are some viruses. We discussed that they have RNA as their genome. Say for example, HIV, human immunodeficiency virus, which causes AIDS in human, which is an RNA virus. So this RNA virus make use of an enzyme that is called reverse transcriptase. Transcription, DNA to RNA is called transcription. What is reverse transcription? Just opposite of that. RNA becomes DNA, and this DNA can now integrate into the host genome and become part of the host genome. And such viruses are called retroviruses, because they enable their RNA genome to be converted into DNA, and they deceive our system, that fool our system. It gets integrated, and our body cannot recognize which is our body DNA or which is the viral DNA. So it will keep propagating along with the cell. And as and when this virus required to transcribe itself, it will make a copy of the RNA from this integrated genome, and it will propagate itself. That is why retroviruses, once they are integrated, it is very difficult to destroy. So we are talking about 4 different enzymes. One is DNA dependent DNA polymerase and DNA dependent RNA polymerase. And there are enzymes which can make a copy of RNA from an RNA, that is RNA dependent RNA polymerase. And then comes reverse transcriptase, that is making a DNA from the RNA. So all 4 possible enzymes contribute some way or the other in the process of called central dogma. And in the translation, when you see, translation is basically conversion of the RNA nucleotides into amino acid sequence. How does it do? As you can see here, there are 3 U's are mentioned here. UUU, which is present in the RNA, which act as a codon, and this brings in phenylalanine. Phenylalanine is an amino acid that is attached onto a tRNA. So that tRNA has got anticodon that can pair with this UUU. That means the anticodon of the phenylalanine bearing tRNA will have AAA, triple A will be there, and during protein synthesis that will come into picture. And then you also have 3 stop codons. Only 3 stop codons are there, starting from bacteria to human. All organisms have got only 3 stop codons. One is UAA, UAG, UGA. They also have names called ochre, amber, opal, like that. And they do not bring in an amino acid, and hence we call it as stop codon. So wherever there is any of these 3 codons are there, you will end up getting a stop codon. And total 61 meaningful codons are there, and these 3 dead stop codons are also there. And total 64 codons are possible, and we will see the codon preference table, etc. or the genetic code, etc. in the subsequent classes. So basically these triple bases are called codons, and every amino acid will have to have 3 bases. Or in other words, if you have an RNA that has a coding region of 300 bases, it can bring in 100 amino acid. Why? Every 3 base brings in 1 amino acid. And for doing this, you need to have ribosomes, which are nothing but ribonucleoproteins. And wherever an amino acid can come in, we call that codon as a sense codon. And where no amino acids can come in, we call it as stop codon. In the right hand side panel, that's what you are seeing, the same what you saw as the part of the central dogma, you can see the second part is nothing but the protein translation, that what we saw so far. And if you look into prokaryotes in the bacteria, the transcription is quite simple compared to a eukaryote, because you have a primary transcript that is to be formed from a given stretch of DNA. So you can see that this is a DNA double-stranded, DNA double helix, what you are having here. And what is shown in this green color is the regulators of transcription. That means it's also a DNA sequence, but it do not code for an mRNA. It has region or area that can help in the recognition of the certain protein or recognition of some elements that will allow where to start the transcription. And then you have a yellow color region, that is the regulation of the translation. That means it forms under some of the UTR untranslated region, etc. And that can influence whether how effectively a mRNA should be translated. Then in the blue color, you have the actual coding region. That means it brings in the amino acid. And then lastly, you have in this deep pink color, the signals for the termination of transcription. So what do we understand? There should be signal for where to start the transcription, and what are the area to be transcribed, and where to stop the transcription. And that is the first step of transcription in prokaryotes such as bacteria, is the production of a primary transcript. And since the bacterial genes are less complex, you end up getting a straightforward functional mRNA. And every mRNA will have a start codon, that is AUG. AUG brings in methionine. Every protein will have methionine as the first amino acid, no matter whether it stays back or not, because in some cases this methionine will be selectively removed. But at the time of production, every protein will have methionine as the first amino acid. So start codon is only one, that is AUG. In the genomic sequence, it will be ATG. If you are talking to a DNA, it will be ATG. So now the second process is the production of the protein, that is protein translation. So the mRNA codons on the mRNA are translated into an amino acid sequence with the help of ribosomes and tRNA, and this mRNA is now getting converted. Getting converted means getting encoded into a protein sequence or an amino acid sequence. And same procedure is followed by retroviruses. The only difference is they have to convert into RNA to DNA, and this DNA is now getting stored in the host, and whenever the host is undergoing transcription, this RNA comes out. This is how it works in the retroviruses. Now, if you look into transcription and translation in eukaryotes, it is very much similar to that of the prokaryotes. The only difference is that they have a nucleus. The nucleus is pretty strong to protect the genome. Pretty strong in the sense it is not available for the cytoplasmic proteins to interact or intervene. So the primary transcript is produced, as in the case of bacteria, from the DNA itself. As you can see here, there is a double-stranded DNA. So the primary RNA has to be modified on both the ends. It has to undergo modifications, and the introns are removed. The intervening regions are also transcribed. They have to be removed for the actual functioning of the mRNA. Otherwise, that mRNA is useless. So after processing, what happens? The mRNA is now exported to the cytoplasm for the translation by the ribosomes. So let us see here. You can see there is a double-stranded DNA here, and it has got regulatory sequence, as in the case of bacteria. So the regulation of transcription and also elements required for regulation of translation also present in the untranslated region part. In a mRNA, it has got a coding part, and also it has got 5' untranslated region and 3' untranslated region. Untranslated itself indicates that it is not translated, it is not getting coded for protein. So you have exon 1, exon 2, exon 3. In between you have intron 1, intron 2, which is present in between these exons. And when they are transcribed, both introns and exons are transcribed. Now it has to be matured. It has to undergo a process, and this process is called RNA splicing. The RNA splicing allows the joining of the exon and removal of the intron. And once the RNA, pre-mRNA is spliced, what happens? The 5' end and the 3' end has to undergo some maturation process. 5' capping, 3' polyadenylation, etc. Once all this process happens successfully, then it will be exported to the cytoplasm. Cytoplasm is the place where the mRNA will find the tRNA, ribosomes, etc. And they help or they work together to make a polypeptide in the cytoplasm. So this is what you should understand about the transcription and translation of prokaryotes and eukaryotes in a simplistic manner. Now let us see what are the types of RNA molecules that are present. So types of RNA molecules, they are messenger RNA that act as the messenger or a message that converts the genetic information into protein. So they stand in between. And then transfer RNA, they are the tRNAs. They help in the production of proteins. And then ribosomal RNA, they also help in the production of proteins. So mRNAs are the intermediates that carry genetic information from the DNA to ribosomes. Transfer RNA or the tRNA, they act as the adapters between amino acids and codons of the mRNA. And then ribosomal RNA, they are basically structural and they also play catalytic components of the ribosomes. This is called central dogma direct. Now let us see what are the other types of RNA molecules that will participate in the indirect way of influencing the production of protein. They are small nuclear RNA, snRNA. They are structural components of the spliceosomes. And then microRNA, they are coming as the short single-stranded RNAs around 20 to 22 nucleotide in length that block the expression of complementary RNAs. And then RNAi, that is RNA interference, that is similar to microRNA, and the double-stranded RNA that is seen in plant also influence the translatability of an mRNA. And siRNA is small interference RNA that also can influence the expressivity of an mRNA. Then comes pRNA, that is PV interacting RNA, normally seen in the germ cells, identified in mouse germ cells, and it's also a non-coding RNA. So what do we understand? These RNA molecules are not influencing the central dogma as such directly, but it can affect indirectly. That means whether you have food at home or not, that indicates whether you are rich enough to have food. But whether you are able to eat or are you willing to eat depends on your willingness to eat plus your body's nutritional status is dependent on by whether or not you eat. That is an indirect way of regulating your nutritional or your health status. So these molecules do not influence the transcription, or they are not able to influence the gene expression in the first step, but they can influence the gene expression in the second step, that is the translation. So an mRNA is there, but whether it should be allowed to express into protein or not is decided by these molecules. So that is why we call these molecules as central dogma indirect. That means they are not going to affect the central dogma direct, but they can influence the central dogma indirectly. So let us look quickly at the central dogma once again. The central dogma in molecular biology is that the genetic information flows from the DNA to DNA. That means the DNA has to make a copy of itself using DNA-dependent DNA polymerase. Otherwise, it will be lost. And during the cell replication or the chromosome replication. And then from DNA to RNA during transcription, because without which gene expression cannot happen. This is also part of central dogma. Then from RNA to protein during translation. So these three steps are the major focus points of the central dogma. All other features that will tweak the system based on which cell or which organism, what condition the cell is living, etc. Accordingly, the central dogma will be tweaked. We can study more about the central dogma in the next class. Thank you.